Hi there, science fans. My name is Dan Casciano, and you are watching Science and Other Stuff for Senior Citizens. This channel is dedicated to promoting science that is understandable to non-scientists and is primarily directed to senior citizens as well as anyone interested in increasing their understanding of science. I would be very pleased if you subscribe to this channel and also click on the bell to be reminded when new episodes are published. Previous episodes have been dedicated to understanding the structure and function of the human cell, human DNA, and the human microbiome. Because of the recent COVID-19 pandemic, I thought I would develop episodes related to the structure and function of that virus, its infectivity, its organ specificity, and other aspects related to its spread and its possible containment. In the present episode, I will be mainly discussing what a virus is and how a vaccine is developed, focusing on different vaccines being considered to stop the infectivity of COVID-19 and eventually killing it. First, we should understand what constitutes a virus. A virus is a small microscopic particle made up of a genome or genetic code that is either RNA or DNA that is surrounded by a protein coat. The accompanying cartoon is an artist's representation of the COVID-19. The protein coat surrounding the genome is a spike glycoprotein. That is, a protein molecule that is connected to a sugar molecule. In addition to several other molecules like a membrane and certain enzymes, its genome is an RNA and not DNA. This genome contains only some of the information needed for replicating or making another virus particle. The virus is dependent upon hijacking the molecular machinery belonging to the host, namely the infected human, to make more viruses. A virus cannot replicate or make another virus on its own. It must infect cells and use the molecular machinery of the host cells to make copies of themselves. Because the virus cannot do this on its own, it is considered a non-living thing and only becomes alive once in the cell of a human. The first cells infected appear to belong to the respiratory tract. Viruses are not bacteria, so antibiotics cannot kill them. And viruses are generally killed by antibodies made by the infected host, or by vaccines, or in some cases can be eliminated through the use of antiviral medications, or by medications that influence the immune response to the invading virus. This slide reminds us of the structure of the COVID-19 viral particle. This virus is spherical in shape with various viral coat proteins, for example, spike glycoproteins and hemagglutinin esterase enzymes on its surface. These molecules can be recognized by the host immune system and could initiate the development of an antibody specific for those proteins and may eventually protect the host from this virus. Also, there is a membrane envelope that surrounds the virus RNA informational molecule. The virus attaches it to a specific site on the surface of the cells and enters the cell through an invagination process, resulting in the eventual shedding of the external viral coat and injection of the RNA informational molecule into the cell's nucleus. Here, various host or cellular molecules allow the synthesis of proteins that eventually constitute the viral coat and the eventual synthesis of more viral informational molecules. The proteins and RNAs are assembled into new viruses and then leave the infected cell by a process of evagination. Thus, large numbers of viruses are produced that can be used to infect new hosts and eventually repeat the cycle. So how does the human body attempt to protect itself from invasion of viruses, bacteria, and other pathogens? Fortunately, the human has an amazing immune system. The immune system defends our bodies against invaders such as viruses, bacteria, and foreign bodies called antigens, and it is composed of special cells, proteins, tissues, and organs. Some of these defenders are special white blood cells, lymphocytes, called B lymphocytes and T lymphocytes. B lymphocytes are produced in the bone marrow, while precursors of T lymphocytes are also produced in the bone marrow, but leave the bone marrow and mature in the thymus. Each B and T lymphocyte is specific for a particular antigen or foreign body. B lymphocytes are triggered to make antibodies. However, certain lymphocytes can destroy the invading virus, while other T lymphocytes are called memory cells, and they can remember the specific invading particle and can destroy them when again infected by them. Since the human has never been exposed to COVID-19 before, we don't have any memory cells to inhibit their infectivity. Therefore, we can only be protected after being infected and make our own antibodies to them or be injected with a harmless vaccine so we can produce our own antibodies to COVID-19. Check out this video about vaccines from the Oxford Vaccine Group. Although this video describes how our immune system responds to a bacterial infection, the biological process is similar to a viral infection.
To understand how vaccines work, it helps to look first at how the immune system works, because vaccines harness the natural activity of your immune system. There are about 100 trillion bacteria and viruses on your body. Not all of them cause disease, but some are able to get inside our bodies to multiply, and this can make us ill. There are barriers to stop this happening, but suppose some disease-causing bacteria do get through. Your immune system is quick to recognize them as invaders. This is because the proteins or sugars on the bacteria's surface have different shapes to any of the ones in the human body. They trigger a complex chain of events involving many different types of white blood cells working together. One type of white blood cell is able to make antibodies to fight the invaders. Antibodies can stick to the proteins or sugars on the bacteria's surface, and this kills the bacteria or disables them. However, not all antibodies will work against these bacteria. They have to be exactly the right shape, a bit like a key fitting a lock. Our bodies have a library of billions of white blood cells, each of which can make just one shape of antibody. Only a few of these antibodies will match the invading bacteria. Producing antibodies of the right shape can take several days. By this time, there could be billions of disease-causing bacteria in your body. Once the right cells are activated, they quickly divide and turn into a production line, making masses of antibodies that stick to the bacteria. Eventually, your body gets rid of all the bacteria and you recover. Antibodies remain in the blood, and some white blood cells may also become memory cells. If those specific bacteria invade the body again, the immune system will respond so quickly that you won't get ill. Vaccines work in the same way. They contain weakened or dead bacteria or viruses, or even just a few proteins or sugars from the surface. This is enough to convince the immune system that a real invader has got in. The same process takes place as when real bacteria or viruses invade our bodies, except you don't get ill. Afterwards, if your body ever meets the real thing, your immune system will remember it and get rid of it before you even know it's there.
What is a vaccine? Vaccines are a type of medicine that trains or instructs the body's immune system to fight a disease it has never been exposed to before, such as the COVID-19. Vaccines are designed to prevent disease rather than treat a disease once an individual has it. There are many ways to make a vaccine. For example, using a viral surface peptide or protein, using the viral genome, the RNA, as in the case of COVID-19, or DNA, essential enzymes, and other viral structures. How does a vaccine work? Vaccines generally work by mimicking viral infections, using inactivated or dead viruses or pieces of a viral protein. These structures or antigens induce the immune system to develop antibodies to that virus. The antibodies made in response to certain viral surface structures inhibit the virus spike protein's ability to attach to the host cell's membrane receptor, thus preventing viral entry into the cell. This results in inactivating the virus. As of July 2020, researchers around the world have developed more than 146 vaccines against COVID-19, and 21 are presently in human testing. More than 125 vaccines are in preclinical studies. 15 vaccines are in phase 1 trials that are evaluating the safety and dosages or amounts to be used in humans, while 10 are in phase 2 trials that fine-tune the vaccine doses that are non-toxic in humans. 4 vaccine candidates are in large-scale efficacy tests that determine whether the safe doses of the vaccine actually can train the human immune system to induce a strong enough antibody response to successfully fend off the COVID-19 virus. And 1 vaccine is approved for limited use. This may be a limited-use vaccine because the antibody response is efficacious but not very strong. This slide summarizes guidelines issued by the U.S. Centers for Disease Control, CDC, that recommend what precautions we should be taking to reduce the probability of COVID-19 infection. They include cleaning and washing your hands often, avoiding contact with sick people, practicing distancing, keeping 6 to 10 feet between you and the other, avoiding crowds outdoors and especially indoors, wearing a mask in public settings and when around people outside of the household. Wearing a mask has been shown to prevent people who have COVID-19 from spreading the virus to others. For more information about science, visit my website at www.dcasciano.com. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comments below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and click the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye. Wearing a mask will prevent those who are infected with COVID-19 from spreading it to others.